A very good evening to all of you who are present here today. This is our July session 2026 South Asia Meetup. We have Vishal Kumar Prasad, who is a very eminent scientist and bacteriologist. And I am going to give a brief introduction of him, and then we are all ears, Vishal. Vishal Kumar Prasad is a secretariat member of the IUCN SSC Amphibian Specialist Group, a PhD researcher at the Laboratory of Animal Behavior and Conservation, Nanjing Forestry University, China, and a research affiliate at the WII. He, his research focuses on amphibian behavior, ecology, conservation, and biogeography, with a particular emphasis on threatened amphibians of the Himalaya and Asia. His work integrates field ecology, bioacoustics, molecular tools, species distribution modeling, and conservation planning to address key questions in amphibian biodiversity and conservation. He has authored more than 45 scientific publications, wow, including papers in leading international journals such as Nature Communications, Nature Reviews, Biodiversity, BMC, and other peer-reviewed journals. He is actively involved in IUCN Red List assessments, conservation planning, and international collaborations aimed at safeguarding amphibian diversity. That's really awesome, and we are lucky to have you for this session, Vijay. So over to you. You can start now. We are all ears.
Thank you so much, Shabni, for giving me this, for inviting me for this talk, and thanks, Carrie, for for making this program. And so I'm very glad that I'm getting this chance to talk about my favorite animals, and I mean my favorite animals on which I'm working for the last almost 5 years in Himalayas. So without like taking more time, I'll come to the talks topic for today. So I'm going to talk about the amphibians, systematics, behavior, conservation in the Himalayas, and mainly in the Western Himalayas, because that's a landscape where I'm focusing more, including the Uttarakhand province in India. And Sabani has already given my introduction, so I'll proceed to the next slide. So, well, it's very interesting, and I would like to ask everyone that why amphibians matter. And I'm sure that most of you must be knowing this, that they are very important for our ecosystem, and they are like barometers. They're like the like very important part of the food web, and they're used for pharmaceutical industry also, like the chemicals from the from the skin of the amphibians is used for making some life-saving drugs for we humans. And also, if there are no amphibians, then this will happen to us. You can see on this image that amphibians play a very crucial role in controlling the pests. And if no frogs, no amphibian on this planet, our quality of life will be so bad, and we won't be able to also survive for long if we do not have the amphibians existing with us. And also, they are pretty good ecosystem engineers in terms of they are also pollinators, as you can see in this. I'll just turn on the pointer. Just a moment. So as you can see in this image that there's a study published in Brazil, and the study shows that the amphibians can pollinate plants. And there are several interesting facts about the amphibians that we all know, so I won't go much in detail, but I'll take you towards the concerns that the amphibians are facing globally. Well, recently published reports and the IUCN Red List assessment, the second global assessment, and also we published the Amphibian Conservation Action Plan in Amphibian Specialist Group, and also some global reviews on the conservation of amphibians. So all these studies point towards a major issue that we need to highlight and that we need to work upon. So 41% of the amphibians are threatened with the risk of extinction at the moment. And I think this number may go high because there are so many data deficient species. And after the red list assessment, there are chances that there'll be more threatened amphibians. And about the Himalaya, this global amphibian hotspot. Well, it's a very unique landscape which has several niches, several micro habitats consisting the high elevation glacial lakes and mid elevation stream where you have the maximum diversity and also the foothills of Himalayas. And also the Himalayas is facing a risk and vulnerability because of a lot of uh, natural hazards that's occurring quite often these in the recent years, like cloudburst, glacial lake outburst that took place in Uttarakhand and Himachal Pradesh and in the Western Himalayas last year, the last few years. And also the glaciers are melting faster than it was expected. They are melting, the warming in Himalayas is more than double than the global average. And we also have several man-made problems for the comfort of humans. We are creating a lot of problems for these amphibians and for these mountains. Like we are creating dams, we are we're going to the like hill stations and we are throwing the waste out there and degrading the habitats of these amazing species. And you can see in the figure a lot of waste is there. Fishes are dying, and you can see that the glacial lake outbursts taking place in these areas. And also, Himalayas is so important because not other than the Arctic and Antarctic, it holds the largest reserve of the ice outside these two poles, Arctic and Antarctic. And it's a very important biodiverse region, which has thousands of endemic species. And from the fresh water that it provides to us, around 2 million people depend on on the water that's coming through the major rivers in Asia. And so protecting Himalayas is not just for the sake of ecosystem or for the biodiversity. It is equally important for our own water security and for the climate resilience and for the livelihoods of millions of people living in this landscape. Please bear with me. There is some issue on my screen. Okay. Well, we have seen that the glaciers are melting faster and these kind of flash floods that took place last year. And now I'll come to my research framework that what me and my colleagues are doing in Western Himalayas. So we are studying the systematics of amphibians in this landscape. We're doing the behavioral ecology studies. We are studying the impact of climate change and working on the conservation. And finally, we are making the policies for the conservation of the biodiversity in Himalayas. So in the systematics, it is very important to first of all know that what species we have, because if we do not if we do not identify the species correctly, then we cannot actually do anything. We cannot understand the species. We cannot develop the conservation plan for the species. And that is why we are doing the integrated taxonomy that combines the morphology studies, like measuring the animals, the holotypes or the specimens, and also we are doing the acoustic studies. We are recording the calls of these vocal amphibians and doing genetics and doing DNA barcoding, and we are coming to a very strong taxonomic framework for the species of Himalayas. Because traditional taxonomy was not complete, and there were several issues with that we're trying to fix. And also, there are more people there working in Himalayas. So collectively, it's important that we follow the integrative approach for the taxonomy. And now let's look at some of the interesting amphibians of the Western Himalayas. Well, we have this Nanorana vicina muripa frog, which is one of the main species in the Western Himalayas, and this plays a very good ecological role in the ecosystem, because the leopard cat feeds on this frog. We have Jonsar torrent frog. I'm sure people living in the Himalayan landscape must have seen this, because they are, if you're crossing the streams, the gadheras, in the evening, in the night, you may hear its call, and you may see this beautiful creature in the night if you're going with the torch. And one of the most beautiful frog of Himalayas, the Amelops formosus. And we have this beautiful frog in the Western Himalayan landscape. We have this Himalayan toad, and we have high altitude specialist toad species in Himalayas. So, and this toad scutiger, it's from the genus scutiger. They are known to occur the highest elevation in the Himalayas. They can occur up to 5,500 meters in the Himalayas, and that's the occurrence of any amphibians found on the highest elevation in the world. So there are so many different families, amphibians of different families that occur in Western Himalayas. We have some more, like Hazara frog. So recently there was some study revision of taxonomy done, and now it's Nanorana hazarensis. It was earlier Alopa, and this is the Iriziolus duduensis that's found in the foothills of Himalayas, and one of the threatened species. It's vulnerable on IUCN Red List. Now let's take some case studies where the taxonomy plays a very crucial role. Well, if you can just like answer from your computer and can just say like which frog do you think it is? Any guesses? Okay, let's just see for a few seconds if someone can give this guess. Like what species is that? Okay, no problem. So I'll go ahead and I'll answer it. But before, I want to just keep this question in your mind, that is this one and this one different species? Well, until now, until we published a paper, they were different species. However, it is not different species. Research revealed that using the integrated taxonomic approach, uh, we found out that it's the same species that occurs in Orissa. The type locality is Orissa in India, and this species occur in Uttarakhand as well. And I'll just give an example like how this I mean, you can yourself see it is different or same by listening to its call. So I'm trying to play the call in the first segment. I hope I can play this call. Please bear with me while I just play this recording. Okay, sorry about the delay. So let's listen to this one. Okay, so this is from near the type locality.
Can you play the call once more?
Yes,
I
I
I
will.
can't hear it.
Okay, no problem. I'll play the call again. And let me know if you hear it.
No.
No.
Oh, really? Because I have many calls to play and like show you like how they are so important.
So while
So shall we?
sharing your PowerPoint, have you picked both audio, video, both?
Well, I just share the screen, Sarvani. Can you just guide me
Could you
through?
please reshare the screen?
Okay, sure, I'll do that. Yeah. So I'll just stop sharing
It will wait. No issues. Yeah.
and I'll share it again.
So in the meantime, Vishal, can I ask a question?
Yes, please.
Yeah, so you mentioned Western Himalayas. So what is the extent of the Western Himalayas? From where it starts and till where it ends in the Himalayas?
Well, sir, here I'm referring to the region that is the eastern side is the up to Nepal, and it or to the west it goes up to the Kashmir and also the Hind Kush, some part of that, and it's mainly on the species distribution that I'm working on, like Nanurana vicina or Nanurana minica and Amelops species that I'm showing. So it might like change a little bit, like people to people, but by and large, that's the landscape that I'm referring.
Thanks.
You're welcome. Okay, so I'll just share the screen. Okay, let's try to play it again. So can you hear this time?
No.
Oops. Well, I'm really sorry. It was meant to work this way. And just give me a moment. I'll just figure out.
You may need to unshare and then reshare, and there should be an option to include sound.
Okay, Carrie, I checked that. Couldn't see it, but anyways, I'll do it again. So let's stop sharing. Oh, okay. Sorry about that. It's been some time that I'm using the Zoom. So, okay. I hope now we can listen to the call. Can you?
Yeah, it's coming through.
Oh, great. That's very wonderful. So let me play it again. So the first call is for the first frog, and the second section is for the second frog. Now the second. So what I want to say here is that though they look different, and there was a study which published the second frog as a different frog. When we studied it, when we checked the acoustics of the two frogs, two species, and also we did the DNA barcoding, we found out that these are not two species, they're just one species. And that was a range extension of the Fajerware Uri census in Uttarakhand. So using this multi-evidence, multiple approach of integrating the morphology, bioacoustics, and the genetics, we could solve this taxonomic uncertainty in this landscape in Uttarakhand. We have one more study that we published in 2022, where the Euphylactus sanophylictus, which is a widespread species that was thought to be occurring in the Shivalik and foothills of Himalayas, but our study revealed that this species is not the Euphylactus sanophylictus. It's a Euphylactus adolphi species, which was described long back in history, and we erected this species again. And now we call it Himachal skittering frog because the type locality of Adolphi was from Himachal. Recently, we published another study in Nature Communication that revealed that in the Dattaphranus melanostictus, which is a widespread species across South Asia and Southeastern Asia, there are multiple lineages, and they were well supported by the mitochondrial as well as the nuclear genes. And we found out that the species that we have in the Himalayas, in Uttarakhand, in Himachal, and in the Kashmir region, that is the lineage which is yet not named. It's unnamed lineage. However, we're just calling it Dattaphranus melanostictus for now, following the frost. But hopefully, in the future, somebody will resolve this taxonomy. And now, coming to the behavioral ecology. And this is one of my favorite and interesting subject. And why? Because it tells you more than the taxonomy. It tells you a lot of hidden information about the species, about their habitat. So I'm more keen to study the breeding biology, the advertisement calls of the species, the release calls. What are these and how come they're different? What is their role? What is the function? I'll be talking more about that in the coming slides. And haptic partitioning that we've studied in some species, and communication, how they defend themselves, and all those things. So let's come to it one by one. So in a case study that we published a few years ago, we found out that these two microhylids, balloon frogs, are calling in the same habitat at the same time. So they're very fossorial species. They come out to call or to breed for a very narrow window for only for a few days, and we were very interested to know how they call and what their breeding behavior. So we recorded the call, and we could find out that they are creating a segregation in their calls as well as in their microhabitat. And let me play some calls, some interesting calls. You might have listened to them. So that's Euperodon cystoma. The other one is Euperodon globulosus. So interestingly, they're calling the same puddle, same habitat, and they're explosive breeders. So there is a chance that the females might not recognize the same species, and there could be a chance of hybridization. So, and then that's not good for the species because hybridization can actually threaten the species existence also. So we studied that how these species, these two syntopic frogs, are managing to occur in the same habitat, calling the same habitat, breeding in the same habitat, and laying eggs in the same habitat. So we studied that, and our acoustic study found out that their call properties are very different. They're significantly different between each other. And also their microhabitat, there's a strong partitioning, significantly different between the two species. Just give me a moment. So this species cystoma calls from the center, from a deep water, and the globulosus calls from the edges. So that's how they are partitioning their behavior, their microhabitat and their calling behavior. And we also demonstrated the vocal distinctiveness of individuals, like how many individuals can be identified based just on the calls. So for these species, we used Beecher's index, and we found out that this frog can be found out around 14 individuals in the habitat, and of course they're not more than 14. So that's how they ensure that they're not hybridizing and they're successfully breeding. And for globuluses, I think the value was more, right? Because they are more common, and yeah. So this was a very like for myself as well, this was a very good learning about the Beecher's index. And people have studied a lot in the birds, but it's rarely studied in amphibians. So, and moving ahead, okay, we published this in 2022. And now there's an interesting interaction between the individuals. And I'll leave it up to you to decide what they're doing. So let's play. Okay, so well, I hope you might have liked this video, and it's so interesting to see that how these uh, individuals are fighting with each other. And I wanted to ask that what do you think about it? What they're doing, and are they all male, or there is a female also, or questions like that. But I guess uh, maybe that'll take more time. So better I just quickly brief you through, as you have seen most of the things. So I came across this amplecting pair in Nainital district in one of the streams in spring, and I found out that there is a huge competition between the Tephranus Himalayanus, that's the species, and the males are competing for the female, and uh, yes, and they were also calling, which I'll be showing like a bit later. But so all I'm trying to say here that there's so many things happening in Himalayas, and probably we do not know about this. And so it's a short video, and maybe some of you can go in the field and make a short note of it. And it's so interesting to document the natural history of the species as well. And so like this,
Sorry.
there were several more examples that I'd be happy to share. I'm sorry, you have a question?
Yes, sir. It's very interesting. And the topmost one, the males are fighting with each other, and female are below that male. Is it okay? Is it correct?
Yes, that's right. I'm sorry, I'm just presuming that some of you are
But
familiar with the
Ishan,
amphibians. So yes,
how
so
they
that's a
behave
female.
like that? Yeah, but how they behave like that? Because the process of genetic transfer is not going on. Just is it just control the female now, or that process is different? Mane, this exchange of or fertilization process later on, or already it has been completed, or it is the just initiation process. Just their bonding is initiated. I'm so sorry. Two, three question is there.
No
Please, sir.
problem. Very, very interesting questions, and I'm happy to answer. Well, they haven't yet started the egg laying. So it is a very initial, phase when this male is clasping the female. Well, it's doing it wrongly because it's a peak season of breeding, and they want to breed and reproduce. So mating has just started, and he's just trying to compete for the female at this moment in this video. And later on, like they lay eggs and the egg strings, and so on, and the mating will go on further.
Thank
I
you, sir.
hope I answered the
Thank you.
question.
Yeah, thank you, sir. Thank you very much.
You're welcome. Well, they have more interesting interactions like this, but maybe next time I'll show more because we have just like 1 hour today. And
So we shall actually continue your talk for next month if it is not, if there is not enough time. Please carry on.
okay,
People will have questions.
sure, certainly. Okay, so from the behavioral ecology, I'll just come back to my interesting research. I am more interested in bioacoustics, and so I want to share some of the findings and some of the vocalizations of the species and talk a bit more about the bioacoustics. But before that, it's very important to know that uh, bioacoustics is a very important technique for the species identification, as we have seen that to correctly identify the species, bioacoustics play a crucial role, especially in the cryptic species. And it's important for monitoring the species. Uh, it helps in the automated surveys as well. Like you have the uh, like audio moth installed, and it's keep on recording the sounds every 5 minutes or 30 minutes, and you don't require manpower to be there all the time. And bioacoustics also helps in detecting rare species. And I'll tell you more examples in coming information. So here, I'm sure all of you will love this frog. It looks green, charismatic, this one. And well, I think one of the most beautiful frog in the Western Himalayas. My personal favorite also. And I was very curious to find out how this species vocalize. And so I could record the calls with my team. And let me play it for you. I'll play once more. Okay, so any guesses what kind of behavior this amphibian is displaying? I mean, if you want, we can do chat.
Related call.
Wonderful. Any other guesses? Okay, so you're very right. It's a stress related, and actually, it's a call when the species is. I mean, this frog was like when there is a threat for predation, like a snake is trying to eat the frog, or the frog is in distress. The frog emits this sound. So, and we can see that here in the this oscillogram and this spectrogram here. Oops. So in this oscillogram, that shows the temporal properties of the call, and this below that, the spectrogram is showing the spectral, the frequencies, and yeah, those properties mainly. So you can see that there's so many harmonics, and this frequency of this call is quite high. It's around 17 kilohertz. We can hear up to 20 kilohertz, and this frog is quite close to that. And I'll play another call. So this call is the distress call of the frog. And they're mainly having the high frequency. Now about the second frog, second toad. Okay, one more time. Okay, any guesses what is this sound? In case anybody.
Territorial.
Okay, we'll try. And anybody else would like to try? Okay, no problem. So you're quite close, but precisely this is the advertisement call of the Dattaphragis himalayanus. And we recorded that in the streams. And the breeding was about to, like, they're advertising their territory, and they're calling the female through the calls, attracting the females. So here we see that there is a huge difference in the spectral properties between the two types of vocalizations. Here we see that the Dattaphragis himalayanus is calling or emitting its advertisement call on a very low frequency. Mainly the dominant frequency would be somewhere here, which is around 1 kilohertz or so, between 1 or 2. And here this distress call is, it's quite like on high frequency, up to 17 kilohertz. So that's a very one of the like difference between these two type of behavior of the species and two different vocalizations that amphibians emit. So I hope you enjoyed listening to the calls, and I'll proceed to the next. Okay, well, those are very interesting sounds and information, but now let's discuss about a case study of one of the frog of Western Himalayas, which has not, which is it lost or is not seen from last several decades? And there are some like speculations that this frog has been seen. However, I tried finding information, but there's no published literature which confirms that this species has been found in its type locality. So this is Amelops chakratensis, chakrata stream frog, and it was described in 1992 and was collected in 1985, so almost like 41 years before it was seen by any human. And we actually tried looking for the species. We surveyed for 2 years in the type locality in Chakrata, and sadly, we could not find even a single individual. And we published this study as the evidence of the local extirpation of this species from the type locality. And so there are a lot of species in Himalayas that may be facing the same crisis. So we need to survey more and more and find these species. That's the holotype of Amelops chakratensis that I took photograph in the museum in ZSI. Well,
Vishal, just a request, could you please explain what a holotype is? Because we have vast audience from different backgrounds.
sure. Yes. Well, this holotype is the specimen that was collected to describe this species. So if someone has to study and compare another individual and say, oh, this is Amelops chakratensis, so the person should have some reference material. So that is the holotype. I hope that is clear.
Thank you. Yes.
You're welcome.
Thanks.
So I can suggest to all of you, including the scientists and researchers, and also some nature enthusiasts, nature lovers, if you're interested to find the species, look in these kind of habitats, like in the streams, and it's actually a rare species. Also, we studied and found out that the IUCN Red List didn't have a proper distribution range for the species, and the type locality was away. So we suggested that the range of the species should be this shown in this red. We also documented several threats to the species and surveyed in the streams in the type locality, and some interesting videos here. So these are the type of habitats where Amelops chakratensis can be found. So I suggest that it's one of the species that may go extinct if we do not, like put efforts to, if we do not take conservation actions to to find this species, to discover it, and then conserve it. Because Himalayas is facing a lot of threats in its type locality. And we also suggest that we should also find this species in and around the type locality and outside. Maybe that we might find it outside the Chakrata. And I recommend that people should publish, and because this species was data deficient until 2023, and recently it was listed as a critically endangered. So if we knew about the species earlier, uh, then we could have tried to save this species. So let's sample it. Let's try to find the species in Uttarakhand. Other than this, we are doing a lot of awareness and capacity building programs because we think that it's equally important to uh, encourage the local students, local communities to tell them, to let them know what biodiversity they have and what incredible species that they have. Beautiful frogs and this lost Amelops frog. So we took lectures in schools, in colleges, and in several government offices as well, and promoting the amphibian conservation in Uttarakhand. And a lot of people are supporting this research, this conservation initiative of finding the Amelops chakratensis and other amphibians occurring in the same landscape. We're thankful to the Forest Department of Uttarakhand for helping out, supporting us in our research. And we're also doing the citizen science programs. So if you see a frog in Uttarakhand, any amphibian species, do take a picture and upload it on the iNaturalist. This will help a lot of scientists to understand the distribution of the species, and maybe someone will see the rare Amelops chakratensis, and they can just put a picture, and it can help to rediscover that species. And in this project, we have 3,000 or less observations, and well, that's not 111, that's less, but it's showing the more areas. But all I'm trying to say that people are getting interested to know more about frogs, and it's a very positive sign for the amphibian conservations in Western Himalayas. Well, we recommend that, especially in Himalayas, if we are sampling, field sampling, we suggest that we should avoid collecting the species. Better to have non-invasive sampling method. You can do a buccal swab, and the frog is alive. You can take the data and leave it back, and of course take permission from the forest department first. And the other way to find the rare and this lost species, we recommend that we should do eDNA sampling, and that can increase the possibilities of finding this species. And whenever we are traveling from one site to another site, we suggest that we should always maintain the hygiene, because we are going from Dehradun to Chakrata or to Nainital or to any higher elevation, let's say Munsi area somewhere. We might carry some diseases in our foot, some bacteria, some viruses that may go to the habitat of the species, and they're not immune to these viruses and bacteria. And also the cuttid fungus or other funguses may travel this way. So the suggestion is that when we are going in the forest, better to just clean our boots, our gum boots, with the ethanol, or just wash them thoroughly with the soap, and make sure that no soap is left in the shoes and not contaminating the water in the stream. So by following these simple practices, we can actually help amphibians and other aquatic biodiversity. This is our research team, and they all have been helping me in my PhD and also in the amphibian conservation. And if you have the passion to save frogs, to save amphibians, let's connect, let's collaborate. And we have a Instagram page for Himalayan amphibians. You can scan it and you can get in touch with us. And you can write to me on my email address. And if you have any query, any suggestion, please feel free to email me. And we would like to thanks and I acknowledge the support of the PCCF office of Uttarakhand Forest Department and ZSI and Lab of Animal Behavior Conservation, Professor Amail Borji, who is my mentor, and Dr. Gupta from Wildlife Institute, and lots of people from the local communities who came with us to the surveys, and a lot of kids. They were very happy to see the frogs in the villages. So thanks to everybody, whoever was a part of this. And I hope that with the collaboration, with the working together with local communities, with the institutions and the scientists, we can protect Himalayas. We can protect the amphibians of Himalayas in a much better way. And Himalayas are very important, as I mentioned, for our water security, and a lot of people depend on Himalayas for their livelihood. So thank you so much for your time. And you say that we cannot conserve what we do not understand, and what we cannot understand, what we do not study. So let us study the amphibians and conserve the amphibians of Western Himalayas. Thank you.
Vishal, your work is excellent, I must say, and very nice to hear all the stories and your efforts and the initiatives uh, you have made to conserve the amphibians, to find the amphibians, to get people involved in your work. That's a very important thing. If you cannot actually involve local people, then probably all your effort will be in pen and paper. But this is a real work you have done, and this is a time for question and answer. So I think our participants will have questions for you. So it's over to the participants.
Thank you, Shivani. And yes, uh, please feel free to ask if you have any questions. With my little bit of knowledge, I'll be happy to answer.
Thank you, Vishal. Wonderful presentation. Uh, so I wanted to know how many species in the Western Himalayas are elevationally restricted. That is, within a band of, say, 5,500, 2,000 meters, uh, not going below that, not going above. How many species are there like that?
Well, sir, it's a very interesting question, and I'm happy that you're answering it. And one of our studies is under review, and that answering your exact question. And we have found that between 1500 to roughly we had 1600, like the band of the elevation actually. So we found that within this 1600 to 2000 and some range, yeah, we have the maximum species occurring in Himalayas. And as in when we go up, we go more than 3000 or above, the species richness decreases. And when we go down, we have many species, the diversity increases, like if you are going below 900 meters or 800 meters. However, you have more of the lowland species there. So the elevation that you pointed out is we have the maximum species of Himalayan endemic amphibians. And in my studies, I would say we have around 20, 21 species that we recorded so far. So we have, I would say, half of them are occurring in that uh, uh, mid elevation zone.
Thank you.
My pleasure.
Any question from anyone? Please
If there
unmute
are,
yourself and ask
can
the
I ask,
question.
can I ask one more question?
Yes, sir, please.
Yes, sir.
See, you said that the maximum number of species are in the middle elevation, and many of them are also endemic and restricted to the Western Himalayas. So my question is, the Himalayas itself is not very old, and many of these lineages have come to occupy there, and they are the species that you have mentioned are representative of multiple lineages,
That's
which
right.
are supposedly endemic to that elevation zone. So, what is the? Are there more niches? For how do you explain the middle elevation being the most important? And considering that the Himalayas has offered both at the latitude and the elevation many niches, how do you say that that region has the maximum number of species in an ecological,
Mhm.
manner? How do you explain it?
Well, sir, because the habitat variables or the habitat factors that are favoring the species uh, are found most in that like in that elevation band, mostly in the mid elevation area. And we have done so I'll just share a part of the study that is under review. So what we found that the habitats, so it's not the elevation actually, these are the habitats. So the species are actually occurring in that particular range because of the habitat. Uh, and so because the habitat is available, the species are found there. So though my research question is slightly different, and I am not 100% expert to answer your question, but yes, I think that because of the more niches available in that habitat, it's neither very extreme, like for the ectotherms, for amphibians, that they are able to breed at least in summers or overwintering. So somehow they are able to they are thriving in that elevation range nicely.
Thank you.
My pleasure.
Vishal, we have another question from Srishti. What features does the high altitude specialist have for them to adapt to such harsh environment where most other amphibians cannot survive?
Wonderful question, and this is one of my PhD research topic, and one of my chapter is based on this work. So we are studying. So that's why also I cannot say a lot of information uh, till I defend my PhD. But yes, it's like for the conservation, I'll definitely answer it. So we have seen that Himalayan topography and the elevation and everything is so complex that comparing it with the plains is like not making much like I would say the good findings in. So I was using a different approach that is good for the like planes, but then I had to change my approach to the boosted regression tree, looking at the elevation complexity in Himalayas, and we're finding that the species are changing their suitable habitat, and like the endemic species are changing the habitat. However, the widespread species are more fit in adapting the changing habitats. So, which hence that uh, like lowland species are also moving up. They are also their suitable habitat is becoming available in the higher elevation, and they are also moving up. And endemic species are also moving up, but then there is a point after that they cannot go ahead because then the suitable habitat is not available for the endemic species. So that is why there was a paper published recently in Nature Climate Change by Murthy, some researcher. I can share it with you. That shows that the species of the alpine region are more threatened with the climate change compared to the species from the other biodiversity hotspots. So that is why it's very important that we study more species in Himalayas, and because the situation for these amphibians is not really good, and it's going to be bad to worse in the emerging threat of climate change.
Thanks. There is another question from Trilo. Can eDNA approach is used in the context of Amelops chakratensis or any other species which is lost from their type localities to get DNA?
Well, eDNA technique is one of the modern techniques that helps identify or like finding out the species which are not present when you're sampling that area. So, well, I don't know like whether we can find it through eDNA or not, but that's one step ahead of the conventional field sampling, right? So having field sampling is very important. Along with it, if we can add to the eDNA sampling, the probability of finding the species goes high.
Thank you. So I had one request. The papers you have shared here, can we have the full paper? Is it possible for you to share
Yeah,
the
sure.
full papers?
I can
Okay,
share those
and I
papers,
can
the
post it
PDFs.
in the group.
Okay,
Yeah, that would be really nice. And
sure,
I will share the papers in the group so that people can have access to the whole paper and can read about it.
sure.
Okay, there is another
So,
question from Dr. Kartik Vasudevan. Did you perform a niche model of Amelops chakratensis?
well, not yet because we do not have the enough uh, occurring points for the species. So it is very difficult for us to, model the suitable habitat of the species in the absence of uh, sufficient occurring points. And that is making the thing more difficult. But hopefully, in coming time, I'm working on a technique which would at least tell us that where we can find Amelops chakratensis, which are the most suitable areas, most likely. It won't be as good as the species distribution modeling, but it'll be at least better to start with that these are the habitats, at least go and check, look for the species there. So in coming time, I'll be happy to work and provide this information.
Just a connected
Okay.
question. How many locations are there of the species?
Well, sir, as for my knowledge, there is only one location in Chakrata. That's a type locality, and that's it. Like where the species occurred in 1985, Dr. Ray had collected from there. Other than
But
that,
you haven't seen in any other place?
no, sir. I haven't seen.
Oh.
Well, there are speculations that it's seen, but I mean, in science we say if it's not published, it does not exist. So it's better that we get some information, some published information for this species.
Vishal,
Yes, sir.
you said like there is one location. Can you just touch upon what is the population size if you have any data? Like this is in continuation to Karthikeyan's first question.
Sir, actually, I do not have the answer, sir, because we do not know the population of the species. I myself have not, I have not seen the species yet. And if it exists, then I need to sample more, and I'm sure like more people are also working. So later we'll have the answer of this question. But as of now, I just know one location from where it was described. So making it difficult to work on it.
Okay, thank you.
I have, may I have some question about? I have only, I have one question. One silly question is there. We have observed
No problem, sir.
migration
Please go ahead.
in
Yeah.
various classes. Okay, fishes, apes, or even in insects, odonata, butterflies. In amphibia, is there any chances of migration, local migrations, or some any you have any informations?
Well, sir, I mean, as far as I think, amphibians are bad at migrating. They're like, they are there. They have a population. They're just there. They're not like tigers or other elephants, like they're migrating so much. There would be a little bit of migration, but they're mostly coming back to breed at their, where they were like uh, to the same breeding spot. So I think they do not migrate much. But uh, connecting to your question, I think that maybe uh, our knowledge is not enough. Like my knowledge, what we publish is not enough, and that's why we recommended that in our paper that we should look outside as well. We should look to sample the species outside the type locality. So maybe
Okay.
that's a hope that uh, with more efforts we can find it.
So you told me just now ki they used to breed in same locality or same places in a same stream or something. How the adult because during summer or that time the stream and the water bodies are near about 90% dried up. So how they came and it is genetically it's it's inherited behavior ki they just return back or they sense the abiotic factors and they return back on that place for moisture food like that.
Well, sir, as far as I know, there are some salamander species in the South America and Central America. They come back where they were like where the like they were hatched, or it's not hatched, but like they were like completed their metamorphosis, and they grow there. They go look for food, but when they have to breed, they come back to the same habitat, to the same locality.
So
So keeping that an example, I'm saying that more or less the amphibians, they come back to the same breeding place when they have to breed.
so second question is for this DNA sequencing and other things. We need a permission from foresters, or if we just go for non-destructive, then uh, we need to contact them or not?
Well, sir, yes, permissions are required, and even for the buccal swab, for example, we are just doing the buccal swab for the DNA, and we have taken the permission from the forest department, and so I mean that's the way to do it. Yes, and collecting samples without the permission of the biodiversity boards of the states or the forest department, I think it's not recommended.
Thank you, sir. And sir, what kind of bioacoustic equipment and software is required for that? This kind of, if we want to go for this acoustic study of these species.
Okay. Well, sir, I go very simple. Sorry, I stopped sharing the screen. I'll connect it again. Well, I use Raven Pro, which is simple to use, and there are different softwares people are using. But for amphibians, for my study, Raven Pro is good, and I can also show you how it looks. And if you're interested, and
Yeah.
I mean, maybe in a week training of a week or so, anybody can learn it and can analyze the calls. So
Thank you, sir.
yes, that's the yeah.
Okay, so we can have
My screen. Can
one
you see
more
my screen?
question. Uh, no.
Oh, okay.
We can take one more question. I thought that Dinesh sir is going to ask one question.
Okay. Okay. Now you can see the screen. Yeah. Yes, Dinesh sir, please go ahead.
No. Like I was just trying to tell, like the last question they were asking. Permission is required to access any biological sample. It can be like tissue, animal DNA, whatever it is. Yeah. And you see that.
Okay, sir.
Okay, thank you, Vishal, for this
Okay,
intriguing
sorry.
evening.
Yeah. So just
And
to show you, in case anybody is interested in the bioacoustic softwares,
Yeah.
it's fun, analyzing calls and playing with it. You can even listen to the calls by playing this. In a click, you listen to the call. You can open the calls and check the each pulse or each note, like to the maximum amplification and things like that.
Okay, sir.
So very easy and interesting to work on this bioacoustic tools.
And recorders?
Okay, so for recorders, I will show you.
I'm sorry.
Well, I use. No,
Able
I'm
to
happy
you.
to answer. Like it's a. Uh, I'll open my slide because there is a one recorder that I use.
Okay, sir.
Just a second. Okay, so if you can see my screen, uh, if you see this in this picture, I am carrying this Sennheiser microphone, and there's a frog here hidden somewhere. Can you see the screen?
Yes, sir.
Okay, and so I'm using Sennheiser microphone. It's a unidirectional phone. It's a bit old, but now we use the Tascam as well, which is not that expensive, which would be like around 20,000 Indian rupees, and you can get the recorder. And another 20,000 rupees or so, you can get the Sennheiser mic. And if your
Okay.
budget is high, then you can even go for higher end products.
Okay, so initially we'll go for some moderate one, then
Okay,
we'll for the things.
sure. So
Thank
if
you
you
so
want to
much.
work on amphibians, you can
Yeah.
just let me know your requirements, and I can suggest you few recorders that
Thank
are
you so
like
much.
not expensive and helpful in the field.
Thank you, Vishal sir, and thank you, Sarmani ma'am, too, because she is the person behind these all shows. Thank you so much, ma'am. Thank you,
Welcome.
sir. Thank you.
Thank you so much.
welcome. Anything for the amphibians. Thank you, Vishal, for such a wonderful evening. And your work inspires a lot. And your work involves a lot of people from research background, scientific background, nature lovers, enthusiasts. And this is the way to conserve uh, the ecosystem and the amphibians actually. So it's really wonderful. And please share the papers and so that I can actually share it with the participants. And if the participants have any more questions, then I'll let you know. You, if you
Okay,
have time, you can answer the questions. I will get back to them with the answer.
sure. Yes, you can also email me in case you have more questions. And I just sent you the paper,
Thank you so much.
and I'll send some more papers quickly in the chat if you allow me just like 10-15 seconds. And if you would like to learn and work together on bioacoustics, I'm happy to collaborate and uh, learn this technique and explore it together.
really wonderful.
Thank you so much, Vishal, for coming here and sharing your knowledge. Thanks to Sarbani for setting this up, and thanks to everyone for showing up. Please come on back next month, last Saturday of every month, same time. And Sarbani, have you shared? You guys have a WhatsApp group? Maybe you can put in that share info in there. And everyone, I put it.
WhatsApp group, right?
Yeah, everyone I put into the chat. There's a another Save the Frogs event happening this Tuesday, 8:30 p.m. IST. We've got Save the Frogs grantee and founder of the Nature Hub Collective in Kenya, Frederick Kioko Kilonzo, giving a presentation about how he trained 150 citizens and helped protect a critically endangered frog in Kenya on the past Save the Frogs Day. 19th annual Save the Frogs Day coming up April 28th, 2027. Why don't you plan a event in your community to educate people in your neighborhood and to help the frogs in your part of the world? SaveTheFrogs.com/day. I am Save the Frogs founder, Dr. Carrie Krieger. And Sarbani, I'll hand it back to you to close this out.
Okay, I have shared the link in chat box. Anyone wants to be a part of Civil Frogs India WhatsApp group is welcome to join the group. We share information and opportunities, and we share knowledge as well. So be a part of the group, and please visit us and please be with us in the next uh, for the next South Asia Meetup session that will be held the last Saturday of the month of August. And thank you, Vishal, again for your presentation. And
Thank you very much,
we will stay in touch.
Sarbani, and thank you, Dr. Kerry, and everyone who participated, who attended the talk today. And I'm very happy to see so many people are interested, and some prominent scientists, Dr. KP Dinesh, Karthik Insan, and many more people attended. So it's an honor to give this talk, and I'm very happy to give this talk and get in touch and
And
let's
there
save
are
the
honor
amphibians.
students as well.
Sorry.
Zoology honor students as well.
Oh wow,
They have
that is so
attended your
good.
talk, kids.
Yeah.
Yeah.
That's so good. I'm very glad. Thank you so much for joining today with me.
Okay. Bye. Good evening.
Bye bye.